Welcome to our exploration of the principles of genetic engineering. In this video, we'll break down the core concepts behind this groundbreaking field of science. Genetic engineering is all about modifying the genetic material of living organisms to achieve desired traits or outcomes. Whether it's improving crop resistance, developing new medicines, or understanding the building blocks of life, genetic engineering plays a vital role in modern biology. Let's start by understanding what genetic engineering really means and why it's so important in today's world. The heart of genetic engineering lies DNA, the molecule that carries genetic information in all living things. DNA is like a blueprint, containing instructions for building and maintaining an organism. By understanding DNA's structure and function, scientists can identify specific genes responsible for certain traits. This knowledge is the foundation for all genetic engineering techniques, as it allows researchers to target and modify genes with precision. Genes are segments of DNA that code for specific proteins, which in turn determine the traits and functions of an organism. Each gene has a unique sequence that acts as a set of instructions for making a particular protein. By altering these sequences, scientists can change how an organism looks, behaves, or responds to its environment. Understanding gene function is crucial for successful genetic engineering. Central dogma of molecular biology describes the flow of genetic information within a cell. It states that DNA is transcribed into RNA, which is then translated into proteins. This process is fundamental to life and is the basis for many genetic engineering techniques. By intervening at different stages of this process, scientists can control which proteins are produced and ultimately the traits of an organism. One of the most important tools in genetic engineering is recombinant DNA technology. This involves combining DNA from different sources to create new genetic combinations. Scientists use enzymes to cut and paste DNA segments, allowing them to insert, delete, or modify genes. This technology has revolutionized medicine, agriculture, and research by enabling the creation of genetically modified organisms with specific traits. Restriction enzymes are molecular scissors used in genetic engineering to cut DNA at specific sequences. These enzymes are essential for isolating genes of interest and for creating recombinant DNA molecules. By recognizing and cutting at precise locations, restriction enzymes allow scientists to manipulate DNA with incredible accuracy, making them a cornerstone of genetic engineering techniques. After DNA has been cut by restriction enzymes, DNA ligase is used to join the DNA fragments together. Think of DNA ligase as molecular glue, sealing the gaps between DNA pieces. This step is crucial for creating recombinant DNA molecules, as it allows scientists to combine genetic material from different sources into a single, continuous strand. Vectors are DNA molecules used to deliver foreign genes into host cells. Common vectors include plasmids, viruses, and artificial chromosomes. Vectors are engineered to carry the gene of interest and ensure its successful integration and expression in the host organism. Choosing the right vector is a key step in any genetic engineering project. Plasmids are small, circular DNA molecules found in bacteria. They are widely used as vectors in genetic engineering because they can replicate independently and carry foreign genes. Scientists insert the gene of interest into a plasmid, which is then introduced into a host cell. The host cell reads the new genetic instructions and produces the desired protein or trait. Gene cloning is the process of making multiple copies of a specific gene. This is achieved by inserting the gene into a vector, such as a plasmid, and allowing it to replicate within a host cell. Gene cloning is essential for studying gene function, producing proteins, and creating genetically modified organisms. It's a fundamental technique in genetic engineering. Transformation is the process of introducing foreign DNA into a host cell. In bacteria, this often involves making the cell membrane permeable so that plasmids carrying new genes can enter. Once inside, the host cell incorporates the new genetic material and begins to express the introduced gene. Transformation is a key step in creating genetically modified organisms. After transformation, not all cells will have successfully taken up the new genetic material. Scientists use selection and screening techniques to identify which cells contain the desired gene. This often involves growing cells on selective media or using markers that indicate successful gene integration. Only the cells with the new gene will survive or show a detectable change. PCR is a powerful technique used to amplify specific DNA sequences. By mimicking the natural process of DNA replication, PCR allows scientists to make millions of copies of a gene in just a few hours. This is essential for analyzing genes, cloning, and many other genetic engineering applications. PCR has revolutionized molecular biology by making DNA analysis faster and more efficient. Gel electrophoresis is a technique used to separate DNA fragments based on their size. By applying an electric current to a gel, DNA fragments move at different speeds, allowing scientists to visualize and analyze genetic material. This method is crucial for verifying the results of genetic engineering experiments and for identifying specific genes. Gene expression refers to the process by which information from a gene is used to produce a functional product, usually a protein. In genetic engineering, controlling gene expression is vital for achieving the desired outcome. Scientists use promoters, enhancers, and other regulatory elements to ensure that the introduced gene is expressed at the right time and in the right amount. Promoters are DNA sequences that control when and where a gene is expressed. By choosing specific promoters, scientists can direct gene expression to certain tissues or developmental stages. Regulatory elements like enhancers and silencers further fine-tune gene activity, allowing precise control over genetic engineering outcomes. Modern genetic engineering relies on advanced gene editing tools like CRISPR-Cas9, TALENs, and zinc finger nucleases. These tools allow scientists to make precise changes to DNA, such as adding, removing, or altering specific genes. Gene editing has opened up new possibilities for treating diseases, improving crops, and understanding genetics at a deeper level. CRISPR-Cas9 is a revolutionary gene editing technology that allows for targeted modifications of DNA. It uses a guide RNA to direct the Cas9 enzyme to a specific DNA sequence where it makes a precise cut. Scientists can then add or remove genetic material at that location. CRISPR is faster, cheaper, and more accurate than previous methods, making it a game changer in genetic engineering. Genetic engineering has transformed medicine by enabling the development of gene therapies, personalized medicine, and the production of important drugs like insulin. By correcting faulty genes or introducing new ones, scientists can treat genetic disorders and improve patient outcomes. The potential for curing diseases at the genetic level is one of the most exciting aspects of this field. In agriculture, genetic engineering is used to create crops that are resistant to pests, diseases, and environmental stresses. Genetically modified crops can have higher yields, improved nutritional content, and reduced need for chemical pesticides. These advancements help ensure food security and sustainability for a growing global population. Genetic engineering also has important environmental applications. Scientists are developing genetically modified organisms that can clean up pollutants, restore damaged ecosystems, and reduce the environmental impact of agriculture. These innovations offer new ways to address some of the world's most pressing environmental challenges. With great power comes great responsibility. Genetic engineering raises important ethical questions about safety, consent, and the potential for unintended consequences. Scientists, policymakers, and the public must work together to ensure that genetic engineering is used responsibly and for the benefit of society as a whole. To ensure safety and ethical use, genetic engineering is subject to strict regulations and oversight. Governments and international organizations set guidelines for research, development, and application of genetic technologies. These regulations help protect human health, the environment, and ensure that new technologies are used appropriately. Public perception of genetic engineering varies widely. Some people are excited about its potential while others have concerns about safety and ethics. Education and transparent communication are key to building trust and understanding between scientists and the public. Open dialogue helps address fears and misconceptions. Like any powerful technology, genetic engineering comes with risks and challenges. These include the possibility of unintended genetic changes, ecological impacts, and ethical dilemmas. Ongoing research and careful monitoring are essential to minimize risks and ensure that genetic engineering is used safely and effectively. Gene therapy is a medical technique that uses genetic engineering to treat or prevent disease. By introducing, removing, or altering genetic material within a patient's cells, gene therapy can correct genetic disorders at their source. This approach holds promise for treating conditions that were previously considered incurable. GMOs are organisms whose genetic material has been altered using genetic engineering techniques. GMOs are used in agriculture, medicine, and research to achieve specific traits such as pest resistance or improved nutrition. While GMOs offer many benefits, they also raise questions about safety, labeling, and environmental impact. Transgenic animals are animals that have had foreign genes inserted into their genomes. These animals are used in research to study gene function, model human diseases, and produce valuable proteins. Transgenic technology has led to breakthroughs in medicine and agriculture, but also requires careful ethical consideration. Gene knockout involves disabling a specific gene to study its function, while gene knock-in adds a new gene at a specific location. These techniques help scientists understand gene roles and develop new treatments. Knockout and knock-in models are essential tools in genetic research and drug development. Gene silencing is a technique used to reduce or eliminate the expression of a specific gene. Methods like RNA interference (RNI) allow scientists to turn off genes temporarily or permanently. Gene silencing is useful for studying gene function and developing therapies for diseases caused by overactive genes. Synthetic biology combines genetic engineering with engineering principles to design and build new biological systems. Scientists can create custom organisms with novel functions, such as bacteria that produce biofuels or medicines. Synthetic biology is expanding the possibilities of what can be achieved with genetic engineering. Gene drives are genetic systems that increase the likelihood of a specific gene being passed on to the next generation. This technology can be used to control populations of pests or disease vectors such as mosquitoes. While gene drives offer powerful solutions, they also raise ecological and ethical concerns. Personalized medicine uses genetic information to tailor treatments to individual patients. By understanding a person's genetic makeup, doctors can choose the most effective therapies and avoid adverse reactions. Genetic engineering is making personalized medicine a reality, improving outcomes and quality of life. Farming is the use of genetically engineered plants or animals to produce pharmaceuticals. By inserting genes that code for therapeutic proteins, scientists can create organisms that manufacture medicines like insulin or antibodies. Farming offers a cost-effective and scalable way to produce important drugs. Bioinformatics combines biology, computer science, and mathematics to analyze genetic data. In genetic engineering, bioinformatics tools help scientists identify genes, predict their functions, and design experiments. The integration of bioinformatics has accelerated discoveries and made genetic engineering more precise. The question of who owns genetic information is complex. Gene patents allow companies or individuals to claim ownership of specific genetic sequences or technologies. This can drive innovation but also raises concerns about access, fairness, and the commercialization of life itself. Genetic engineering is a global effort, with scientists from around the world collaborating on research and development. International partnerships help share knowledge, resources, and ensure that advances benefit all of humanity. Collaboration is key to solving complex challenges and advancing the field. Future of genetic engineering is full of promise. Advances in technology, deeper understanding of genetics, and new applications are on the horizon. From curing diseases to feeding the world, genetic engineering will continue to shape our lives in profound ways. Staying informed and engaged is essential as this field evolves. As genetic engineering becomes more widespread, education and training are more important than ever. Scientists, healthcare professionals, and the public need to understand the principles, benefits, and risks of genetic engineering. Investing in education ensures that society can make informed decisions about the use of this powerful technology. Effective communication is essential for the responsible development and use of genetic engineering. Scientists must share their findings clearly and honestly, while the public should be encouraged to ask questions and express concerns. Open communication builds trust and helps guide ethical decision-making. Golden rice is a genetically engineered crop designed to combat vitamin A deficiency in developing countries. By inserting genes that produce beta-carotene, scientists created rice that provides essential nutrients. Golden rice is a powerful example of how genetic engineering can address global health challenges. Before genetic engineering, insulin for diabetes treatment was extracted from animals. Today, genetically engineered bacteria produce human insulin, making it safer and more accessible. This breakthrough has improved the lives of millions and demonstrates the real-world impact of genetic engineering. BT crops are genetically engineered to produce a protein from the bacterium Bacillus thuringiensis, which is toxic to certain pests. This reduces the need for chemical pesticides and increases crop yields. BT crops are widely used in agriculture and highlight the benefits and debates surrounding GMOs. Gene therapy has achieved remarkable successes, such as treating inherited blindness and certain immune disorders. By delivering healthy copies of genes to patients, doctors can restore function and improve quality of life. These breakthroughs show the transformative potential of genetic engineering in medicine. In conclusion, the principles of genetic engineering have revolutionized science, medicine, and agriculture. As technology advances, the possibilities will only grow. By understanding the core concepts, benefits, and challenges, we can harness the power of genetic engineering for the greater good. The future is bright, and genetic engineering will continue to shape our world in exciting ways.